Thanks for the opportunity to talk to you and for the invitation. So I'm going to talk to you today about a high-throughput phenotyping pipeline we have developed in collaboration with Jim Henderson, who is the UFM wheat breeder, to deliver more productive and stress-resilient wheat varieties. So the key idea here is that we're focusing on the wheat canopy. So why we are focusing on the wheat canopy? It's essentially the engine that drives the yield, and there are several reasons for this. So what plants do, especially domesticated crops, is that they're very good at capturing light. So literally inside the leaves, you have a little antenna that captures those photons to do the work. There are small pores on the surface of the leaves that open to allow for carbon dioxide to enter, and that carbon dioxide builds the plant and builds the yield. Those same pores are the pores through which the plant transpires to cool itself. And on top of that, by cooling itself, the water moving through the canopy generates tension on the water column, which brings nutrients, especially mobile nutrients that Lindsay just mentioned, like nitrogen. So that explains why the canopy is very important, because it's responsible for energy capture, carbon capture, water, and nitrogen, which are key components for yield. Right? So I picked this up from a zoom on this image here, and this is what typically I think Jim sees in his field, which is different canopies from different cultivars. And the key question here is, how do you know which one's working hard for you? So our inspiration here comes from infrared thermography, and you probably know in this day and age what these are used for. And actually, before being used for health, they were used for performance. So many athletes are being tested in settings like this, where you put athletes on treadmills and you get them to run and you image them using infrared thermography. In that way, you can distinguish which one has the best physiology when it comes to doing work. So here, for instance, we have an underperforming athlete whose body is warmer, as you can see with the color scale here. Proportion of yellow to red color over the body compared to this athlete who is transpiring, maintaining processes at optimal temperature because he's cooling himself. So when it comes to a canopy, a cooler canopy means that it is able to really maintain those processes of light, energy interception, carbon dioxide fixation, taking up water by the root, and taking up nutrient at good or even higher rates compared to a warm canopy, because all these processes are really temperature dependent. These are biological processes like us. So if the temperature gets a little too high, they slow down. All right, so in my program, we use thermography as a tool to help agronomists and breeders understand what's going on and develop management and cultivars that are more adapted to certain stresses. And this is a slide just to illustrate the techniques, the technologies that we use. Typically, we use ground truthing, which is very important. This is a kind of buzzword, but it's an important thing. This is essentially consists of using thermocouples to really give this the impression, the sense of how plants themselves are feeling the heat, literally. And then we have kind of remote sensing tools here. We have obviously the drone, on which we have the thermal camera, which takes snapshots like this, more on this later. And on days where flights are not possible, which are typically very windy days, we can use sometimes those handheld cameras. All right. So this technique was validated in different projects. This was conducted on oats over a 3-year study. This is again a zoom on the larger size of the previous picture. This is the entire trial. This is currently under peer review, and the key message is represented here in this figure here. I'm trying to guide you through the information. Here you have the drone canopy temperature in degrees Celsius. So here it's cold, here it's warm. And this is the floret fertility, which is in this specific trial is a good indicator of yield. There's a strong correlation between yield and floret fertility. These are the names of the breeding lines, and what you can see here is that we have a statistically significant negative relationship between the canopy temperature and the floret fertility, which tells us that the cooler your canopy, the higher your floret fertility, the higher your yield. So that, again, this confirms this our premise. But this is unknown. So we wanted to look at this question when it comes to wheat. And this is where our research objective was to develop this technology to conduct high-throughput screening for wheat canopy health and productivity, and ideally identify and capture favorable gene and integrate them in view of a wheat breeding program. And for this, we have two strategies, which are important to highlight here, just to show that the technical kind of wrestling that we need to do to really have good quality data, which is to fine-tune the technology to really help to minimize the experimental environmental noise, because this is way more than just scanning your field and getting the data and be happy with it. You have to filter out a lot of noise and quantify the extent of genotypic variability in these physiological signatures. So we conducted the first experiment to test this idea. And here the question was simple: can we detect differences between lazy and hard-working canopies using this method? In the case of wheat, we validated an oat. Wheat is a different beast. So in this case, we used two groups of 22 lines, very similar to each other, and with the only difference that each group of 22 harbor a lazy or hard-working version of a photosynthesis gene, or should I say QTL, but it's for the sake of the example called gene, identified based on research effort previously funded by the Minnesota Research Promotion Council. We had 3 replications for this trial for each genotype, planted in single rows, and we used 3 drone flights. So these are the 2 guys who conducted this. Just a snapshot to illustrate what the data looks like, at least visually. These are the single rows. This is very high resolution, so each pixel here is 0.15 square inch. And here you have the color gradient within its relationship with the temperature. So this is typically cool single row. This is warm single row, right? So just to understand how this looks like. Now, thank you, Lindsay, for explaining the bucks and whiskers thing. So so it's the same concept. So we have here, I'm going to plot the bucks and whiskers representing the data observed in terms of canopy temperature, here again in Celsius, because that's the format for publication. And these are the outputs for the lazy guys. So this is what the data looks like. So we have a statistically significant difference between the lazy and the hard-working in the right direction, meaning that the hard-working guys have cooler canopy temperatures. This is based on a flight that took place on June 12th. And the same pattern repeats itself on another day and another day. So this confirmed for us 2 things: that technique is working and that it's able to discriminate individuals with slightly different genetic background, which is a good thing. So after this validation, the next step for us was, okay, so we're going to try to work in a breeding framework here, so we have a scaling issue to handle. So are we able to scale this approach for a much bigger number of genotypes, moving from 44 to 500 or more? Much higher number of like 25 on-yield trial plots, which are bigger, obviously, plots. So the shift in data size is just visually illustrated here to give you a sense of how much data we have to grapple with to do those types of analyses. So here the genetic material is 504 breeding lines with 4 checks planted in a sample campus by James team. The experimental design was an augmented incomplete block design with 4 checks replicated 14 times. So the total here is 506 times. So it's a huge number for us. We flew 28 times over 14 days using 2 cameras, one RGB, which is red, green, and blue. It's basically camera that you guys know, and infrared camera as well. Used 54 ground control points, which are anchors that are GPS tagged to help us stitch the image together. Thermocouples for ground truthing, as I explained, and a whole bunch of corrections for changes in temperature, relative humidity conditions, altitude during flight, because these things happen, GPS coordinates, which are different between the drone and the GCPs, and canopy height, because those guys keep growing over time, you have to correct for that. All right, so listen, you can look on the video, just to give you a sense of how this thing lays out in the field. So this is the team on a given day, which is June 8th, And this is just the output from the RGB camera on the drone, which is facing downwards, so it goes in a certain direction, and reaches the end of the trial, that goes the other way, and does that zig and zag motion over the entire trial. You can see sometimes here white circles, if you can see them, which are the ground control points here, for instance. The next video will show you the equivalent of this in the infrared. So it's much faster, so for the sake of time. This is not military grade, but still very, very high definition. And it's basically the same idea. If you get late, it's not a problem. So what we do then is we arrange those thermal and RGB photos in a flight path. So here in this example, the drone starts from here, move this way, and zigzag across the entire trial. You see two positions here. The blue one is the initial position. The green was the one corrected based on GPS discrepancies between the drone and the ground control point. So this just gives you a sense of the type of corrections we do. And then we stitch them together to have this type of rendering here. So this is the kind of output that we have. So this is for day, May 26, flight number 14. This is a cold day. This is the color-coded temperature here, this time in Fahrenheit. And what we can say as an image like this is that the pipeline is able to reconstruct the thermal environment of the entire trial. So this is, for instance, for another day, which is actually the same day where those videos were taken, flight number 27, June 8th. And you see it's warmer because it's a warmer day. And you can already start to see hints of differences in color between the clouds, which for the computer are very easy to detect. For the human eye, it's not that easy. So for the sake of the example, in the next slide, we change this color scale to really highlight the difference. So please don't pay attention to the yellow here, but essentially here what I want to draw your attention to is the difference in shade or in color gradient. I'm a little bit of a colorblind person, so I'm trying to help you. To highlight differences between genotypes, because that's what the end game is. And in this case, we're focusing on genotypes which we have multiple replicates, which are the check cultivars. So what you can clearly see here that in this day, there is MN Torchi, which is warmer compared to Washburn. So you see temperatures here of 95.2. It's higher actually than the air temperature. 94.8, etc. This one here is much cooler. So this means that this guy, he's opening small pores, moving water around, dumping water in the atmosphere, cooling itself. The other one is stressed and shutting down its stomates. So this is one day, and this is a specific example. Washburn was cooler by 9 degrees Fahrenheit compared to Torchi, and ended up out-yielding it by 6 bushels per acre, which is quite significant, up to 12% difference. But this is just for sample, more applications are needed. So if you integrate all replications, so in this case we have not the 3 but the 14 replications averaged here, and you include all the days processed so far, because I've shown you the example for 1 day, now we have 7 days. You have this tendency here, which is consistent with our premise, meaning that the cooler the genotype, the higher yield performance. So I mentioned the 500 lines. The data is still being processed. This is kind of the slide for the same day. Here on the y-axis, this is the temperature in Fahrenheit. Here, these are the 504 entries. I don't have the space to name them all, but these are like 504 different temperatures at that single day. And what you can see here clearly is that there's a huge genotypic range in temperatures. So 15 degrees Fahrenheit difference. You have a cool genotype, which is much cooler than the air there, 7 or 8 degrees cooler, and the other one is much hotter. Again, the same idea. One is opening its pores, doing its job. The other one is stressed and closing its pores. So this is, again, promising finding and opens up opportunities to continue in this direction. So I mentioned yield plots just for the sake of the exercise. We have large variability again in yields with 80 breeding lines out of those 504, out-yielding the best performing check by up to 16 bushels per acre. These are here. These are replicated only once, so the second year was going to be second replication, and third year will be the third one. And that's when we have definite view on the yield potential of these guys. So moving forward, going back to objective 1 and 2, we would like to finish the processing pipeline by this winter and replicate the trial in the spring and summer of 2022. Obviously, this is a lot of work and it could not be done without a really complex team effort. In my team, I have Jose Lopez, who's the lead of the image analysis pipeline, who's now replaced by Daniel Mons, and Vishal, who was involved in the first experiment. And of course, Jim Anderson and his team, who was involved in everything related to this trial, from planting, plant husbandry, harvest, remote management, and everything else. So, obviously, I would like to acknowledge your support in conducting this research, without which we cannot have those interest in the results. So thanks again, and the UofM for their continuous support as well. So thanks again for the opportunity, and please feel free to ask any question.
Thank you, Walid. Do we have any questions from the audience?
Yeah.
This was done down at St. Paul campus.
Yeah.
Is there any correlation with stroke further north? How's it going to compare?
That's
Is
it.
that up down the road?
Yeah, that's a very good question. We would like to do those trials in other locations in order for us to capture other environmental conditions. We know that St. Paul is kind of some sort of a heat island and has its own kind of climate. So for sure, the idea is to deploy this strategy across multiple locations.
Can you physically see the difference on leaf material on these different varieties that you can then relate back to your cool or your warm?
So other than the temperature, it's very hard, like visually, and this is where our initial premise, so this is where the idea comes from, because sometimes there's a difference in greenness and maybe, but these things, at least in terms of the physiology, do not relate to this kind of transpiration thing. So perhaps you could see differences in just the droopiness and the shape of the leaf, but these are not connected to the transpiration. The best way to connect to the canopy cooling is to measure the temperature. So, but the good insight, we had that idea before, through experiments we discovered. So,
are you advocating for higher seeding rates to achieve better canopy?
Seeding rates? I think the canopy performance has more to do with the genetic potential of the individual rather than with the management can help. So it's maybe not seeding rate, maybe nitrogen applications will boost the canopy a little bit. It is known to boost photosynthesis. Not sure about the seeding rate though.
I have a question. You had the comparison with Porgy and yes, Washburn.
Yeah.
Okay. How did I don't have my data in front of me. How did they compare in this year's trials? In other words, we had Porgy in our trials. Porgy looked pretty good.
Yeah.
Did it translate to a higher yield for Washburn in the trials?
We talked about this with Jim, so he can follow up with an answer. But so in this trial here, so again, this was a kind of a droughty year for sure, and especially in Minneapolis and in St. Paul. Torgy, which was slightly warmer, ended up being slightly underperforming in that specific environment. But Jim is going to say how Torgy has performed.
Yeah, great. Torgy did about 4% better than Washburn statewide, and even another trial that was adjacent to this trial, Torgy did about 10% better. So this was a little bit of an oddball, but I think it does show that the cooler canopy temperature does result in a higher yield. So that's kind of the takeaway for me.
Thank you.
I think we have time for one more question if there's one out there. I have one last question before you leave. Is there any way to take the information about canopy conductance and canopy temperature and maybe backwards infer any information about drought tolerance in specific varieties?
Yeah, it's a good question. So The canopy temperature is a good indicator of, could be used as a good indicator for drought tolerance strategies. So Washburn here was able to be a water, so obviously it's cooler, so meaning it's spending a lot of water, right? So a guy like this, maybe in an environment where the plant, the wheat is grown on stored soil moisture, which is drought-prone environment, this strategy might be end up being costly, and because it's a risk-taking approach. In contrast, Storgy, who's not kind of shutting down its pores and saving water, might end up being actually outperforming Washburn in a drought-prone environment because of it has a water-saving strategy. So we can use this type of information to, at least conceptually, and we can do that assisted by modeling, to know where to deploy certain varieties based on a canopy temperature, which we use as a proxy for estimating how aggressively they use water.
All right, thank you, Walid.
Oh, thanks.